Okay. Good afternoon. Can you hear me? Yes. Can you hear me? Send me a response. Yes. Okay. All right. Okay. So by now, you need to know we are starting the discussion at 5 because we have a full paper, full four structured essays to discuss. Okay. Can you hear and see me clearly? Any video, audio issues? There's an issue with my voice, I know, because I am not recovering exactly. I'm still under the effect of a probably viral or something. Okay, probably not dengue virus, but let's see. So you guys have to be safe from the dengue virus, particularly. Okay, if you have been infected before, double careful. If you haven't been infected, don't get it. Make a parakvat hadaganna ඕන කරන disease එකක් නෙවෙයි. So be protect yourself from mosquitoes. Okay, especially in the daytime. All right. Okay, so we'll slowly move on to the discussion. Some people might join later. Okay, we'll slowly move on to the discussion. So all of you have gotten your marks by now, I think, online and physical both, right? You might have received your marks by now. Okay, so on while the discussion is happening, you can interact with me, you can argue, you can say anything that is different from my marking scheme or my answer. So I'm open to that. Okay, because these are model questions, these are not made according to exact exam definite marking scheme such as in a past paper. So it's all up to logical sort of reasoning. Okay, but what is the ultimate reference point? It's going to be your resource book. So we are doing all this so that we can practice the content in the resource book. Okay, so I'm open to all the, let's say, any other opinions that you have in the, so keep the chat active and ask any doubts, anything that you want to clarify. Haridh? Okay. Okay, we'll start with the SEQ questions. Okay, what is the most abundant group of biological molecules on earth? Yeah, what is a group, right? Group of biological molecules, easy, very easy things. Okay, so it's going to be carbohydrates, right? Name the nitrogen containing structural polymer found in the exoskeleton of some animals, right? So what is the animal phylum that we are talking about? Arthropods, right? Arthropod exoskeleton is composed of chitin, no? Chitinous exoskeleton. Okay. Then next question. Okay. Name a reducing okay. Right. Rejoining the class. Okay. You're very welcome. Yeah, nothing has changed. We are doing classes at the same time. The paper discussions are happening on Fridays. Okay, and for physical people who are in the class, I'll be switching to a physical paper class. I mean, so the online paper class is happening at the same time. I'll be doing a physical paper classes at ISM. I'm still looking for a slot arranging with the institute. So I'll probably try to keep it on Friday. Okay, so I'll let you know. So you can join physically with the paper discussion as well. So, okay, so once that starts, you can properly write papers at ISM and then properly join the discussions as well. Okay, right. Name a reducing disaccharide. You have several options. Neither you have several options. Not several. You have two options. You can say maybe maltose. Everything other than sucrose. Maltose versus lactose, right? Sorry, maltose and lactose. Okay. So name the non-reducing disaccharide. Okay. So you can just say sucrose. Who is the remaining one? This is the famous one you need to remember, no? Right? This is the famous one you need to remember. Okay. Okay, question number 4 in the space given below. Show using suitable diagrams how a peptide bond is formed between two amino acid molecules. Right. Whether there was no the space was not given for the drawing. Okay, so I hope you managed it. Now I am facing the same problem, so I'll try to manage it here. Right? How do you draw the formation of a peptide bond? Okay, so you need to first draw two amino acids. So I'll start with the central carbon, and there's a C double bond OH, and there's a hydrogen, and there's a variable R group, and there's a NH2. Right? This is just one amino acid. Okay, so just for clarity, we'll call it R1 so that you can say it is a two different amino acids are joining. Okay, then we can add another amino acid with the same orientation, C hydrogen. I'll just draw it as C double OH. Okay, C double OH, and there's a NH2 as NH2, and let's say R2. Right? Okay, so these are the two amino acids that are going to join, and so this is the peptide bond formation. What's going to be the interaction? The OH will provide, I mean, the COH group will provide the OH group, okay, and the amino group will give one of the hydrogens. Okay, so you are going to have, let's say, we'll start with this carbon, and this is the R1. Okay, this is R1, this is hydrogen, CO, and this is the peptide bond. Okay, and there's a nitrogen with just one hydrogen now remaining, and carbon, hydrogen, COOH, and we'll say this is R2 for the second group, and this is NH2. Okay, and what is something very important that you have to indicate, there's a water molecule that is going out as in the form of H2O. Okay, so okay, the marks allocation, sorry, right? So for previous carbohydrate 1, chitin 1, for any one of these, there's 1 mark for 1 mark, and marks allocation for this, for the two amino acids both, I will give only 1 mark. For the drawing of two amino acids, I will give 1 mark. You are releasing one hydrogen, I will give 1 mark. Okay, you are releasing one hydrogen, I'll give, sorry, one water molecule, I'll give you 1 mark. And for saying this is a condensation reaction, okay, I will give 1 mark. And for the final, this entire structure, if you have drawn it correctly, there's 1 mark. And also for this labeling of the peptide bond, you have to label this as peptide bond, I will give another 1 mark. So altogether for this question, draw using suitable diagrams, I'm going to give 5 marks. Okay. Okay. Altogether for that, totally 5 marks. Okay. Then, what is the test used to detect the presence of peptide bonds in proteins? Peptide bonds in proteins. Okay, right. It's the Biuret test. Say the Biuret test. KOH and copper sulfate dala, we detect the presence of peptide bonds. So if you joined with us very early on, you might remember from the practical seminar we did. Had, I mean, we had the Biuret reagent, commercially produced Biuret reagent was available, but but if not, you can do it with copper sulfate and potassium hydroxide, right? So you can access, I mean, if you have just some free time, you can go watch the practical seminar I did for the 2028 people. It's available in my YouTube channel. Okay. So for saying Biuret test, 1 mark. What is a glycosidic bond? What is a glycosidic bond? So I expect you to write 2 points, right? So the complete answer, you can say a glycosidic bond is formed by the removal of a water molecule by a from two adjacent monosaccharides by a condensation or a dehydration reaction. Okay, so you can say it is a bond formed by removal of a water molecule, removal of a water molecule, that is going to get you 1 mark, by a condensation, or you can say a dehydration reaction. So for saying it is there's a removal of a water molecule, I'm going to give 1 mark. It's either by a condensation or a dehydration reaction, I'm going to give 1 mark. Okay. Name 2 biological compounds containing glycosidic bonds. You can mention biological compounds, no? So you can mention any monosaccharide, sorry, any disaccharide or polysaccharide, right? Any disaccharide or polysaccharide. That is not the answer, Harida. Okay, that is not the answer. I'm just saying you can mention anything: sucrose, lactose, maltose, indala, cellulose, glycogen, starch, right? Whatever you can write. Any any disaccharide or polysaccharide you can write as a compounds containing glycosidic bond. Okay. Any questions up to that? So 2 marks for that. Any two? I will give 2 marks here. Everyone understands the formation of the peptide bond, right? This right? The two amino acids. The OH will be given by the carboxyl group, and the hydrogen will be given by the amino group. There's a condensation reaction. Therefore, there's a water molecule that is being released. I will give 1 mark for that water molecule, and then you have to label the peptide bond as well. Okay. What are the 3 major chemical constituents of a nucleotide? Nucleotide. Basically, what you have a pentose sugar with the oxygen here, and there's a nitrogenous base and a phosphate group. You done, no? Carbon 1, 2, 3, 4, and the 5th carbon is the one where the phosphate is connected. Is a basic nucleotide, right? So write down pentose sugar. Okay, online people, I hope you have received the marked PDFs. Okay, phosphate group, right? So you have received the marked PDFs. So I think many of you received physical papers as well, right? Physical papers were delivered to you at as well. So pentose sugar, phosphate group, and nitrogenous base. Okay, so 3 marks. Okay, 3 marks. Name 3 nucleotides and state 3 functions of each of them. Haridha, let me know if I'm going too fast, but because these are basic early stuff, I'm going to go a bit quickly because we need to discuss 4 structured essay questions. Name 3 nucleotides and state 1 function of each of them. Right. So you have a list now at the end of nucleic acids. As you can remember, we had ATP, NAD plus, NADP plus, FAD. Right. Those things you can write. So you can write ATP, NAD plus, NADP plus. Yeah. What else? FAD. Those things you can write now. So what other things have you written? What other things you have written? ATP, universal energy carrier. Where you have to be careful, is when you are talking about NAD. When you are talking about NAD, you have to say it's an oxidizing agent during respiration. NAD, because you know NADH is found in your cellular respiration. in glycolysis, in Krebs cycle, all those places you can find NADH. So NAD plus is a oxidizing agent in respiration. Okay, so similarly, NADP plus is a oxidizing agent in photosynthesis. Okay, මේ දෙක පටල ගන්න එපා. Other than that, all the other functions are similar. So in photosynthesis, of course, you know NADPH නේ තියෙන්නේ, right? At near the uh, photosystem 1, there's the synthesis of NADPH, and here you can also say it's also found in respiration. So what are the other common things? May tunatama common name. What are the other common things you can write for NAD plus, NADP plus, FAD, coenzyme, electron carrier, right? So you can write they act as, those are the safe things to write. I'm here mataka nattang a bit, right? If you can't remember between difference between NAD plus and NADP plus, right? What are the safe things to write? You can say electron carrier, right? You can just say they act as electron carrier, or you can say coenzyme, right? Those are the safe options, right? So for writing at least so the 3 nucleotides 3 marks and the 3 functions correctly 3 marks, so altogether 6 marks. Are okay, moving on. Say the benefits of systematic classification in the study of organisms. Okay, we are moving on to a bit of evolution. The part B. Okay, so at least 5 points have to be written here. Okay, what are the 5 things you can write? Yeah, systematic classification. Okay, yeah, what can you say? They show evolutionary relationships. You can show evolutionary relationships among different organisms. Then what else? Yeah, prediction of characteristics of organisms based on their groups, right? You can predict. Okay, if you are given an organism in a certain phylum or a class, based on the common characteristics of that, we can say, okay, this organism is likely to have these things, right? So you can predict characteristics. You can predict characteristics of organisms. Then there's a universal system of naming, right? Without confusion, right? Universal system of naming without confusion. Then you can identify accurately because they are properly described, right? You can identify accurately. And finally, it facilitates scientific study. Facilitates scientific study. Okay, those are the benefits of having a systematic classification. Okay, right. Shall we move on? What are the molecular level criteria used in the classification of organisms? Right. Molecular level criteria. What have you written? Molecular level criteria. Yes. So in the present system of classification, we use mainly. I mean, so you have actually five criteria. No? So out of that criteria, what are the molecular level criteria that you can say? What are the molecular level criteria? So you can say a sequence of bases of DNA of important genes. Yeah, that you can take. Then DNA, sequence of bases of DNA of mitochondria and chloroplast. Yeah, those you can take. Then base sequence of ribosomal RNA. Base sequence of rRNA. Okay. Then sequence of amino acids. Amino acids in some common proteins. Then molecular structure of some cellular components. Of some of cellular components. Okay. So for writing all five, I can give you yeah 5 marks. Previously also 5 marks, then not 5. Here also 5. Okay. Sequences basis of DNA of important genes, sequences of basis of DNA of mitochondria and chloroplast, base sequence of ribosomal RNA, amino acids in common proteins, and the molecular structure of cellular components. Because these are very reliable, right? For example, when you have discovered, okay, you think you discovered a new organism. Okay, as a scientist, you are working in the Amazon jungle or whatever, and in the deep sea or something, you think you discovered a new organism. Okay, how are you going to catalog it? How are you going to enter it into the sort of the international catalog of species? You have to explore all these. You have to explore, you have to sequence the DNA of their important genes. You have to sequence the DNA of the mitochondria of the organism you found, right? So if you found in the sort of the international database, if you find some other organisms with having the same data, that means you didn't find a new organism. Right? That means you didn't find a new organism, because the organisms, for example, bacteria, those kind of things can have slight morphological differences. Right? So you think you found a new one, but when you compare it with already available information, okay, you find out, okay, it has already been discovered again, right? Because you can't lie, you can't, these gene sequences, mitochondrial sequences, RNA sequences, they are going to remain mostly the same, right? Other than, so their morphology or the appearance can be different, but these things don't change much. Yeah. Arrange the major taxa used in the classification of organism in the organism increasing number of common characteristics. So, okay, now tell me, should you go from which direction should you go? कोई पता नहीं होता है ना? Is it domain to species or species to domain? Yeah, what do you think? Which direction should you go if you want to increase the number of common characteristics? Yes. Yes, exactly. Hold on. Yes, you have to go from, yeah, top to bottom, because as you know, the number of organisms increase when you go to a higher taxa, high, or increase the hierarchy. But if you want common characteristics, you have to come down, right? You have to come down. So ඔක්කොම ටික ලියන්න තමයි තියෙන්නේ. So domain, kingdom, phylum, class, order, yeah, family, මටත් වෙලාවට අමතක වෙනවා, right? Family, genus, species. Okay, kingdom, domain, kingdom, phylum, class, order, family, genus, species. Right? ඔක්කොම හරියට ලියලා තියෙනවා නම් if you have written all correctly, just 1 mark. Okay. Okay. You have written all correctly, just 1 mark. Okay. This is basic stuff. Okay. Right. What are the two events unique to meiosis that contribute to the production of genetic variation in daughter cells? Yes. These are very sort of a sensible question because in your syllabus they have given only two things that they say that is happening in meiosis that is going to give rise to production of genetic variation. What are the two things? Okay, now I want you to tell me the exact phase of meiosis as well. Right? What is the first thing? That is the sort of the main maker of variation. That is the main incident that is going to produce a lot of variation. What is that? Yeah? What is it happening? Yeah? Crossing over. If you have written recombination, that's good. Crossing over and recombination. Yeah, that happens in Liandone. That happens in which phase? Meiosis 1 or meiosis 2? Meiosis 1. Prophase 1. Meiosis 1, prophase 1. What is the other thing? Independent assortment. Independent assortment. So I hope you can remember what you mean by independent assortment. Can anyone remember what is the meaning of this independent assortment? Because I took a lot of effort to explain this to the 28 people. So I hope you can remember what you know about independent assortment. What do you mean by independent assortment? Yeah. So you know, I'm going to remind karlenna. Okay. So you know, if this is the cell, okay, can anyone tell me in which phase of meiosis independent assortment of chromosomes, homologous chromosomes, okay, තව ටිකක් add කරා, happens in what phase? Yes, anybody? Okay, මම තව hint එකක් දෙන්නම්කෝ. මෙන්න මෙහෙම එකක් ඇඳහම තව පොඩ්ඩක් මතක් වෙනවා. හරහට මෙහෙම එකක්. What am I drawing like this? What is this? I think make a label කරාම anybody can mention. What is this? So the midpoint. Yes. Metaphase plate. Metaphase plate එකක් තියෙනවා නම් is it anything other phase? No, it is the metaphase. Yes. හරිද? So okay, I'll draw a bit of main අපේ කවුද? Main අපේ centrosomes. These are your centrosomes. Okay. So then I'll draw the පොඩ්ඩක් අඳින්නම්, right? So let's say this cell has this is a 4 chromosome cell. This is 4 chromosomal cell. So what do I mean by independent assortment? What do I mean by independent assortment? Is that, so if the red color ගන්න maternal විදියට, take the red color chromosomes as maternal and the blue color as paternal. Okay. Take the red color as maternal and blue color as paternal. And what has happened here? So there has been crossing over. So these red chromosomes have a bit of, there can be a bit of blue. Right? So these red chromosomes can have a bit of blue because of crossing over. And the blue chromosomes can have a bit of red because of crossing over. That happens in prophase 1. So they have undergone crossing over. What do I mean by independent assortment? Okay, is that there's nothing determining exactly if the two reds are going to be on the same side, if the two blues are going to be on the same side, or if they are going to be sort of mixed up like this. There's nothing determining if the paternal and maternal chromosomes arrange I mean, there's nothing determining how maternal and paternal chromosomes arrange on the metaphase 1, metaphase 1 plate. Okay, so there could be a mechanism. We haven't discovered it yet. Okay, so what do we say? Okay, it happens randomly. So that is one of the ways where there can be variation. Can you understand? So both reds can be on the same side, both blues can be on the same side. But now imagine that is this happening in your body cells. In your body cells, how many chromosomes do you have? In your body cells, how many chromosomes do you have? Yeah, you have 46. So 46 means how many pairs? 23 pairs of homologous chromosomes are going to arrange on this metaphase plate. Yeah, when one of your cells are undergoing meiosis, right? So when that happens, there's nothing determining uh, which side is going to get most of the maternal, which side is going to get most of the paternal. Okay, so this is what we call as independent assortment. That is one of the ways in meiosis you get variation. Independent assortment. Okay. Okay, where is my PDF? Oh yeah, okay, right. Understood. So 1 mark for each, 2 marks. Okay, 2 marks. Then, at what stage of cell division does each of the following occur, Haridha? Right. So cell division, bit of cell cycle knowledge, replication of chromosomes, the doubling or the copying of chromosomes. Right. So this is not exactly correct. The chromosomes I would like if it's replication of DNA because it is the DNA that is being copied, and then the DNA will wrap around histones to form chromatin. Okay. So anyway, that happens in which phase? Yeah. S phase of interphase. Okay. Division of the centromere. That means the centromeres separate. Okay, there's division of centromere. There's a small issue there. Okay, now tell me, what are the answers you wrote for this? I didn't exactly notice this. What are the answers you wrote for this? Question B. Yeah. What are the answers you wrote? Kianba? Yeah. What are the answers you wrote for this? So actually, what I wanted to ask here was, so division of centromere actually happens in anaphase. Separation at the centromere happens during anaphase. Am I right? Okay. So when that happens, I mean, so you hands up if you have written anaphase as the answer for this. If you have written anaphase as the answer for this. Okay. Right. Anaphase, I think it has been marked wrong. Yeah. So you can take a mark for that. Okay. If you have written anaphase. Okay. So there was the question is supposed to be duplication of centrosomes. The question was supposed to be duplication of centrosomes. That was the question, proper question. So hands up if anyone wrote G2. If you wrote G2, anyone who wrote G2? Hands up. Anyone wrote G2? Yes or no? Yes? Anyone who wrote G2 for this? Okay, then you can take G2 as a correct answer. I think G2 has been given the correct in the marking process. But if you have written anaphase also, you can take a mark because this says centromere. No? So, division of centromere, you know, separation of the sisters at the centromere happens during anaphase. So, you can take anaphase or G2. Both are correct. Okay. Don't think about it. Just learn theory right now. So, S second 1 mark. This is 1 mark. Okay. Arrangement of chromosomes on the equatorial plate. So, equatorial plate is not in your syllabus, but it's you can understand it is the metaphase plate. No, why do you call it equatorial plate? Because it's sort of somewhat similar to your equator. Eh? If you think the cell is the Earth, then this is kind of like the equator, right? It's kind of like the equator. So you can call the metaphase plate as the equatorial plate as well. So you can understand that's what we are asking. Arrangement of chromosomes on the metaphase plate. So that's why they haven't given the metaphase plate. You can anyone can write the answer is metaphase. So it happens in metaphase. Yes. Okay. Reformation of nuclear membrane or nuclear envelope that happens in yeah telophase. Telophase. Okay. Right. First structure is a question is over. Any doubts? Any doubts? Let me know if you have any doubts. Okay. Okay. We'll quickly move on to the second structure, is it? Okay. 2A1. What is the gas release in the light reactions of photosynthesis? How is the light reaction? Now recall. I will, as we go along, I'll remind you as well. In the light reactions, what is the gas released? It is oxygen gas because of the breaking down of water molecule. Okay. What is the source of this gas? H2O. Water I'll give you 1 mark. Two major factors that affect photosynthesis. Okay, there are several. Okay, right? Light intensity. Of course, you know photosynthesis, light, CO2 concentration, temperature, water availability, then pollutants. May are very bit unlikely they can bit. I can give you marks, but of course, I prefer if you have written CO2, light intensity, temperature. Those things are the best ones. Then inhibitors also you can write. Inhibitors. So any two, I'm going to give 2 marks. So 1, 1, 2. Okay. Yeah. Nothing. So any two is fine. Name the two products of light reactions that are used for the synthesis of carbohydrates in the dark reactions. Right. What are the two outputs of your light reaction? So the light reaction actually fixes energy so that energy can be used to fix what? The actual output, that is food, no organic food. We consider usually it to be glucose. Okay. So the light reaction is the energy harvest. Okay. You harvest energy, you store it in certain molecules, and that molecules will be used to transfer that energy into the Calvin cycle where the actual food production happens. Okay, so what are the two products? You have the direct energy molecule, that is ATP, and you have your electron carrier, that is NADPH. ATP is the main energy source, NADPH is the electron source. Okay. What is the function of RUBP carboxylase, right? So it is like RUBP carboxylase කියලා කියන්නේ right අර එයාගේ professional name එක නේ. He is not saying to anyone I am also known as oxygenase, because then it's not good ne? Because he's catalyzing the oxygenase reaction as well other than the carboxylase. So Rubisco means RUBP carboxylase oxygenase no, but මෙතන දීලා තියෙන්නේ RUBP carboxylase. So who is that? අපි දන්නවා අපෝර කවුද කියලා, right? It's Rubisco, right? අර හම්බෙද්දි කියන්නේ okay, right. What is the function of RUBP carboxylase enzyme in photosynthesis? So you need to write a small sentence. You have to say it catalyzes. This is a very important word. It catalyzes the addition. It catalyzes the addition of CO2 to RUBP. That's enough. Okay. Catalyze the addition of carbon dioxide to RUBP. 1 mark. Within 2 marks. ATP and NADPH. Sorry, I have given 4 marks for this. Yes. Within the dekai I have given 4 marks for the 2 products of light reaction. Where is this enzyme located? I want the precise catalyzes the fixation of CO2 by RUBP. Yeah, I can give you marks, but you can phrase it better, right? It catalyzes the addition of CO2 to RUBP. No, so by RUBP it seems like the RUBP is doing the fixation, right? RUBP is just there to catch the carbon dioxide. The catalyzation, I mean, it is done by the Rubisco. So where is this enzyme located? Where is the Calvin cycle happening? That is where the Rubisco is located. Where is the Calvin cycle happening? Is it in the your thylakoid membrane? Then chloroplast එකක් you know, there are stacks of thylakoid that we call as what? Grana. So grana are connected by intergranal lamellae. That is where light reaction happens. But is it so where is the Calvin cycle happening? Calvin cycle happens in the matrix, no? Calvin cycle happens in this matrix of mitochondria. So you have to say, I'm sorry, stroma of chloroplast. Okay, right? Matrix of mitochondria, but not here. Okay, stroma of chloroplast. Chloroplast is stroma. Okay, stroma of chloroplast. Matrix is there in mitochondria. Okay, stroma of chloroplast. Okay, right. We'll move on to part B. State the concepts of the cell theory. ABCDs වගේ මතක තියෙනවනේ, A for apple, B for ball වගේ. What are the 3 parts of the cell theory? I'll just keep say it, okay? All cells are composed of, all organisms are composed of cells. Cell is the basic structural and functional unit of life. All cells arise from pre-existing cells. Cell is the, all organisms are composed of cells. Cell is the basic structural and functional unit of life. All cells arise from pre-existing cells. Okay, marks. But then for saying this stroma of chloroplast, 1 mark. Cell theory got 3 marks. What are the internal structural features of eukaryotic cells that are okay different from those of prokaryotic cells? Bit of a internal structural features of eukaryotic cells that are different from prokaryotic cells. Okay, what have you written for this? Yeah. So I think the marking scheme was a bit strict on this, right? Structural features, you can write what are the things? DNA. So they are asking eukaryotic cells and how they are different from the prokaryotic cells. So what are the things you can write? In case of genetic material, you can say DNA is linear and found in the nucleus. That is a feature of eukaryotic cells. Then DNA associated with histone. That is one another thing you can add there. Then you can talk about ribosomes. Both 70s, 80s ribosomes are found. Okay. Then you can say membrane bound organelles are present. Membrane bound organelles are present. What else you can say? You can talk about flagella, right? Flagella complex with 9 plus 2 arrangement, and they are intracellular. The flagella is intracellular. Okay, those kind of things you can write. Yes. Yeah, I think organized nucleus better, right? This is good enough. DNA is linear and found inside a nucleus. So that means you have a nucleus that sort of implies that there's a nucleus. Okay. Okay, a student was provided with the epidermal peel of an onion mounted on a glass slide and a light microscope, stating the correct sequence of steps that should be followed to observe the shape of the onion epidermal cells under the light microscope. Okay, Chutikali Karapudeval, I think I demonstrated this to you in the practical seminar under microscopes practical. So I'll just read the answer to you. Then what are things you have to do? Uh, So the student is already provided with the epidermal peel and it is mounted on the glass slide. So you don't have to say using a razor blade, you obtain a thin slice and you put it on a petri dish with some water. Then you take it, you pick it up with a paintbrush and then you put a drop of water on the glass slide and then you put the onion peel on top of that. Then you cover with a cover slip without wrapping air bubbles. Do not need any of those because you are already given a prepared glass slide and a specimen. Okay, so now you have to say, you have to follow all the microscope steps, right? All the microscope steps. So you place the slide on the stage. Okay, so that is the first thing. You place the slide on the stage. Then you observe under low power. Of course, this is the next one. You observe first under low power. Then you can you move the slide to bring it to the position of study. Then adjust the light source, right? That is next. To give optimum illumination. That is, if you have written optimum illumination, that's good. Okay. Then you have to talk about the sort of the order of the focusing knobs. What is the first knob you are going to use? Cos focusing knob to improve get the image as clear as possible. So you make the image as clear as possible by the coarse focus. Then only you touch the fine focus, right? So I think I have told you this. In some microscopes, there's a mechanism. Okay, when you are moving the coarse focus, okay, you move the coarse focus and you make the image as clear as possible. Then there's a way to lock the coarse focusing knob. There's a way to lock, so you can push it in or pull it out so that it becomes locked. Okay, then in most microscopes, in some microscopes, you can only rotate the fine focusing knob if you have locked the coarse focus. And if you have locked the coarse focus now, you can move the fine focus. That is because you have to finish your coarse focusing before you move on to fine focusing. You have to finish your coarse focusing before you move on to fine focusing. And also for exam settings, then for example, if you are given a microscope for an exam and you have a time limit, usually they have done the course focusing for you, right? Course focusing already done. So they have locked the course focusing knob, and you are only able to move the fine focusing knob. Because why? When the next person comes to do the look at the microscope in the exam, that person has to do the focusing from the beginning. Course එකත් කරලා fine එකත් කරනවා. So because that is disadvantages, there's a way to fix the course focusing knob. Anyway, so you use the fine focusing knob to improve the quality. Then finally you can add next step, you can add rotate the nose piece. Nose piece කියන එකට I will give 1 mark. Nose piece කියන එකට to bring medium power and then high power. Okay, so these are the marks distribution. I have given 8 marks for this. Okay, stage කියන එකට 1 mark, low power කියන එකට 1 mark. Then to give optimum illumination or adjust the light source, any I would have given 1 mark. Coarse focus 1 mark to get the image clear as possible. Fine focusing to improve the quality. Then you rotate the nose piece. Okay, so 8 marks for this question. Okay, lowest magnification per point. Okay, adjust the level. I would prefer if you use the low power low power objective lens, low power objective lens. Okay, that is the power that is given there, no, because it is the objective lens are labeled 4x, sorry, times 4 times 10 times 40, right? So they are indicated in the form of their power. Okay, so better use that. And also the so you say adjust the stage doesn't move. Okay, usually the stage doesn't move. The sort of, we say, the lens apparatus or the body of the microscope moves up and down. Okay, observe it through mid power followed by high power. Yeah, sure. I won't give marks for saying mid power and high power. What I'm going to give marks for saying rotate the nose piece to bring other lenses, other objective lenses into use. Hurry. Okay, shall we move on to this question? Part C. Identify. Who is this? Who is this? Lesson ලියන්න ඕනේ, right? Root එකක් කියලා මදි, right? I need a nice full answer, right? Okay. So there are 3 things you should have written here. Okay. So it is the primary structure you need to say, okay, because from this X-shaped xylem and those things, it's a primary structure of a root. Yes, you have to identify, but it's a dicot root. It's a dicot root. Okay, yeah. Be very careful when you are answering the structured essay question. Okay, be complete as possible. Make your answer very complete. Then nobody can argue. So I'm going to give 2 marks. So saying primary structure, it's a dicot root, 2 marks. Hurry. Then you have to do a bit of identification. PQRS. P. Who is P? P is a xylem. Q between the arms of xylem. In between arms of xylem, who do you have? Phloem. I would prefer, I mean, I didn't give you wrong for this. Okay, if you have written primary xylem and primary phloem, I kind of like that. Okay, then I'm sorry, Q is primary phloem. R. Who is R? R is your is endodermis, because immediately after your cortex, who do you have? This is the cortex. You encounter the endodermis. Right? After the cortex, you come into contact with the endodermis. Endodermis is the sort of the checkpoint, from whatever that is coming from the cortex will be filtered by the endodermis. Then S is kind of inner to the R, that is your pericycle. 4 marks. Okay. Okay, label this. Okay, so right, the next question I was going to planning to give all, but because safranine staining is not anymore in your syllabus, no, right? It's no longer there, but just keep in your mind the answer, right? You safranine staining, you can do safranine staining for lignin, okay, for lignified tissues, okay. So it actually stains most of the lignified tissue. So what is the most lignified tissue in this above diagram? It is this our star shaped xylem, our star shaped xylem. So that will be stained by safranine, but you can take all for that, okay. Don't think about it. It has, it's not in your new resource, okay. For coarse focus and fine focus words, only if they are separate points. I don't understand the question. So in this, yeah, for just writing the words coarse focusing knob, using the coarse focusing knob, get the image as clear as possible. That is one sentence. So for the phrase coarse focusing knob, it's going to be 1 mark. You what do you do it? What do you do with that? You make the image clear as possible. Then using the fine focus, 1 mark. You improve the quality, another mark. Okay. Okay. Draw and label a few cells of tissue R. Method that the space is a problem. Tissue R when it is at matured stage, right? So who is R? Okay. R is endodermis. Right. What have you drawn? Yes. Yes. So Mehmai, right? For this, so even though we say okay, endodermis has the Casparian strip, I'm not expecting you to draw the exact Casparian strip containing that sort of a diagram. Okay, so I'm not expecting that from you. What I'm expecting is actually, because it is the innermost layer of cortex. So endodermis belongs to cortex, and cortex is mainly composed of parenchyma. Okay, so cortex is mainly composed of parenchyma. So if you can draw, if you have drawn some parenchyma cells, that's all I expect. So in parenchyma, you have this sort of roughly spherical shape, Okay, then you have a big central vacuole, right? You have a big central vacuole like this, and you have a nucleus that is sort of in the in the pushed to a corner. Okay, then the labeling, you can label this as large central vacuole. Then there's a intracellular space. Okay, then thin flexible cell wall, then nucleus. Okay, any doubts? So, okay, you are supposed to draw cells of the cortex. So, cortex is mainly parenchyma, right? Cortex is mainly parenchyma. So, few parenchyma cells. Okay, let's move on. Right. What are the features of gymnosperms that enable them to be more successful terrestrial habitats than bryophytes? So, there's no competition, no, right? It's like you are comparing. I mean, gymnosperms. That means you have developed vascular systems, and then you have developed seeds as well. The only the thing is you are not doing having flowers, and you are not producing fruits, so that the dispersal is much easier. But other than that, compared to your bryophytes, that means non-vascular seedless plants, the gymnosperms are so advanced, right? So there's no competition actually. Compare right? But that means you can list down a lot of things, right? So you can say well developed vascular system. Okay, well developed vascular system. True roots, stems, leaves. Then you can say production of seeds. Anything else you have written? Mm, be more successful. Sporophyte dominant. That doesn't, okay. So dominance of sporophyte doesn't have much of an advantage, right? Having the sporophyte as the dominant. That is just how the evolution happens. Supporting tissues. You can say, okay, you can say well developed reproductive structures, well developed reproductive structures. That you can say. Yeah. Reduction of gametophytes. Again, so the sporophyte dominance and reduction of the gametophyte didn't allow them to be exactly sort of the to be successful, right? Terrestrial habitats. So for terrestrial habitats, what are the things you need? You need a good dispersal method such as seeds, and you need a good transport method such as vascular tissues, and you need a good support such as roots, stems, and also a good way to do photosynthesis. Okay, such as your leaves. Those are the most important things. Okay, shall we move on to question number 3? Any questions up until now? Going too fast? We have a small part of the MCQ as well, neither. Yeah, any questions? Okay. Which question answer? This one. Yeah. Okay. Question number 3. We have a few MCQ questions to discuss as well, right? Okay. Name a living plant tissue. Okay, 3A1. Name a living plant tissue that provides support. So, what are they implying, right? Okay. So, most of the, use xylem tissue is the main provider of support, and then you have sclerenchyma and tissue to provide support. But what is the only living plant tissue that can provide a bit of support, particularly to a leaf petioles? Yeah, and also you are sort of this sort of herbaceous plants. Yeah, yeah, your kalenkaima neda. Chuti chuti plants also with because it can become turgid with the water intake, and you can provide some support. Name two substances that are present in the cell walls in the tissue named in one above other than cellulose. The maker, this question was given to confuse you a little bit. So if you can't remember exactly the features of cholenchyma, some students might have written lignin and suberin blah blah. Never. Because why? Cholenchyma is still a living tissue. If you have lignin or suberin, whatever, that is going to not allow this plant tissue to live anymore. Okay, so if you need to be alive, you cannot have ligninosovereign. So you can say hemicellulose, or you can say pectin. Yeah, that means what? If you have developed a secondary cell wall that is containing ligninosovereign, can you be a living cell anymore? No, you are going to be killed. Okay. What is parthenocarpy? Carpy, right? Carpy refers to the sort of the fruit, right? Right? That means the development of a fruit without fertilization. No? Okay. So carp is something. It's a word that we use to describe the fruit. So development of fruits without the fertilization. So you can say fruits develop from ovary. Right? Very careful when you write without fertilization. Fruits develop from the ovary because you need to remember it's the ovary that will become the fruit later. Yes, very good. Okay, so 1 mark. Yeah, previously may they got 2 marks. 1 mark. What is parthenogenesis? Then Genesis is something like the birth, right? In the Bible, the Genesis is there. Sort of Genesis chapter is like the birth. Okay, so Genesis refers to the birth. Okay, so parthenogenesis is like the occurrence of life without fertilization. So you can say seeds development without fertilization. Development of seeds without fertilization, you can say, okay, something wrong with the plate. Okay, without fertilization, you can say it is parthenogenesis. Okay, Describe seed germination. Okay, so we need to write the stepwise process of seed germination. Once a fertilized seed or a viable seed, that's a living seed, has dispersed to a favorable area or a chosen habitat, how does it begin its life? How does it begin its life? Are the people not boring, right? Okay, I know you have to. I mean, it's online and but you have to get used to it. Okay, because you might understand this is much more time saving, particularly paper discussions. Okay, so what is the first thing? What is the first barrier that you need to overcome as a seed? There's a in sort of a self inhibition. There's a self inhibition. What is the self inhibition? That means the cell is sorry, the seed is uh, sort of there's a self inhibition. Yes, you have to break seed dormancy first, or you can say overcome seed dormancy. That's the first thing you need to do. What do you mean by seed dormancy? That is sort of the inherent inhibition of the seed germination until all the conditions are favorable. Okay, then absorption of water. Absorption of water, this activates enzymes. Activation of enzymes. Enzymes are the machines, no? So the work is done inside the seed to produce all the new tissues. The machinery is the enzyme. So you need to wake up the machines. Then where they patang and we are about to begin a new life. Okay, activation of enzymes. Then, so if you can imagine the enzymes as the engines, you need to provide fuel to the engines, enzymes. So you mobilize food sources, mobilization, right? Starch with the or bit of lipids with the So if you are a peanut, you have a lot of lipids, yes, stored. Then if you are a starchy seed, maybe rice or a cosattack, right? You have starch stored, right? Mobilization of food sources, right? So if you, I mean, in plants you have glyoxysomes that convert fatty acids into sugars. So those kind of glyoxysomes start to activate, and then so you can say rapid growth occurs. You extend a radicle through the seed coat. So the radicle is usually the first process to come out. Radical extends through the seed coat. Then radical shows what? Positive geotropism, because radical will later become this root. Okay, so how do you draw like a typical germination? If this is the earth and there's a seed, there's a seed that is sort of somewhat like this. What is the first process that will grow out of it? That is the radical, but that radical will go down like this. The radical will go into the soil because it is showing positive geotropism. It grows towards the ground. Then later, choti choti mehema plant leaves again, that will become the plumule. Okay, so you can say plumule shows negative geotropism. Okay, plumule shows negative geotropism. Radical extends to the seed coat and it shows positive geotropism, and plumule shows negative geotropism. That means it grows up towards the sky. Okay, so seed dormancy 1 part, absorption of water 1 mark, activation of enzymes 1 mark, mobilization of food resources 1 mark, radical extends through the seed coat 1 mark, radical shows positive geotropism 1 mark, plumule shows negative geotropism 1, 2, 3, 4, 5, 6, 7. Okay, I will give separate marks for the plumule and for saying radical another mark. Okay, so altogether 9 marks for this question. I'll read again. The seed break, seed dormancy 1 mark. Absorption of water activates enzymes, that is 2 marks actually. Then mobilization of food resources, 1 mark. Radical extends to the seed coat, that is 2 marks. Radical, 1 mark, and it extends to the seed coat. Radical shows positive geotropism, 1 mark, and plumule shows negative. Sorry, radical shows positive geotropism, 1 mark. Plumule shows negative geotropism, 2 marks. 2 marks. Okay. Okay. Right. Part B, name the path that can be seen in a longitudinal section of an unfertilized ovule of an angiosperm. Okay. I'll longitudinal section of a mature unfertilized ovule of an angiosperm. Yeah. So it has to be a mature ovule. Either. So in a mature ovule, what do you have actually inside? What do you have actually inside? I think I can show you my famous diagram of the embryo sac, right? The famous mature ovule diagram, right? Do you want me to draw it or do you want me to show it? Aridha? Okay, so they're asking you to just to name the parts too, right? What are things you can see in the embryo sac diagram, the famous embryo sac diagram? Like a famous scale, don't be afraid of it, right? Something you have done several times. So, how does the embryo sac look like? Hmm. Main, what are things that you have? Yes, going from outside, yeah, going from outside the outermost layer, you have integuments. Okay, then I would give mark for writing micropyle. Okay, so there's 9 things to be written. Okay, so there's a opening here, that is the micropyle. Okay, then the nucellus tissue is there, nucellus tissue, right? Okay, right. Yeah, nucellus is giving nourishment to the female gametophyte or the angiosperm, sorry, the embryo sac. Okay, so nucellus is there, and what else? So you can say the embryo sac is there. Then you have one egg cell, synergids. How many synergids? 2 synergids, 3 antipodals, 2 polar nuclei, and 1 central cell. For all those things, okay. And I think many of you have forgotten to mention the nucellus, right? The nucellus is the remaining megasporangial tissue, right? How did you get a megasporangial tissue? When they made it, this is how the ovary looks like, and inside one of the ovary, inside the ovary, you have ovules. Those are called immature ovules, and in that immature ovule, what do you have? You have a megaspore mother cell. That megaspore mother cell divides by meiosis because that is part of the megasporangium. Megasporangium is the reproductive structure of the female. I mean, it is the female reproductive structure, sporangium. Okay, there's going to be a megaspore mother cell. It is going to divide by meiosis and produce 4 megaspores. Those 4 megaspores, out of the 4, only one is going to remain active, and it is going to divide, it is going to develop into the embryo sac or the female gametophyte. The remaining megasporangial tissue is going to remain as the nucellus, which will provide nourishment to the female gametophyte or the embryo sac. Okay, so those are the things you have to mention. So integument, micropyle, nucellus, then embryo sac. Okay, so there's 4 there. Then you have to mention all the other cells. What are the cells? One egg, synergids, polar nuclei, central cell, antipodal. Okay, so 9 things you can write. So 9 marks. Yeah. Okay. Next, list the post-fertilization changes that take place in the major structures of the angiosperm ovary. Right. So you know, after becoming pregnant, what are the changes that happens to the uh, female gametophyte? Yeah, yeah. Mega excel the over mega keyword. That's fine. Yeah, no worries. Okay. Post-fertilization changes. So what's going to happen? අපි තියෙනවනේ අර ලිස්ට් එකක් දැන් pregnant mother ගේ බඩ ලොකු වෙනවා, she gets morning sickness and feet swells up, she can't see eat anything, right? ඔය වගේ there are things that happens to the the ovary, inside the ovary, because all the seeds have been fertilized. So what's going to happen? Ovules become start to be called as the seeds. Sorry, ovule become seed. Okay, ovule become seed. Then inside the ovule, what do you have? A zygote, no? So the zygote is the first single cell, no? It becomes an embryo. Then, then what happens to the newly generated triploid nucleus? Then, for example, so you know there's something called double fertilization happening, no? So in the method, there's something called double fertilization because one sperm is going to, of course, combine with the sperm's the egg cell, so the egg nucleus, and one sperm nucleus is going to combine with the polar nuclei. So there's going to be a triploid nucleus here, ne? In the middle, in the central cell, there's going to be a triploid nucleus. What happens to that triploid nucleus? That will remain as what? Endosperm. Main endosperm. Endosperm is 3N. The 3N endosperm is going to remain. That endosperm is the source of nourishment or the nutrient source for the developing embryo. So, ekat liyan done here. You have to write that here. So, the triploid nucleus becomes the endosperm. Ovary becomes the fruit, right? So what my point is, do not write half answers. Okay, do not write half answers. I'm training you to do that. Do not write half answers. Then you might come home, okay, after the structured essay paper, okay, I have done well in the structured essay, but you have not, because you haven't written what you know completely. Okay. Then ovary wall becomes the pericarp. Ovary wall becomes the pericarp. Ovule becomes a seed, zygote becomes the embryo, triploid nucleus becomes the endosperm, ovary becomes a fruit, the ovary wall becomes the pericarp. Okay, fruit take pot, right? If you think this is a mango seed, right? Around the mango seed, you have the mango fruit, and the mango peel is the pericarp. Okay. Labeling Kaktin. Identify the two vascular tissues shown in the diagram. Right. So who is this? You can toggle identify. P is who? P is xylem. Q is phloem. How do I identify? One thing you can see these are xylem vessel elements. And also here you can clearly identify when we G කියන man. G is clearly a companion cell. G is clearly a companion cell. So based on those things, you can come to the conclusion. අපි මෙතනම label කරමු. So A is what? A is the end plate. අර නිකන් මේ have you seen in the corner of your bathroom there's a drainage sort of holes, set of holes, so that all your bath water can go out, right? So it's like that, some set of holes. So we call it perforation plate. So it's very, I mean, it is designed so that all the xylem can go flow through it easily. Perforation plate. B are what? B, they are pits. Pits. C, you can say C is a vessel element. C is a xylem vessel element. D, what are D? D, as they have label mentioned, okay. D, have you written tracheids? Anyone who's written tracheids? Yeah, yeah. You can take a mark for that. Okay. Anyone who wrote tracheids, you can take a mark. But I think I gave marks for saying pits. Okay. Tracheids and pits, both you can take a mark. Okay. The best answer is to call them as tracheids. Okay. They are tracheids because they are thin and with sort of pointed ends. Okay. They are tracheids. Okay. Then E is a xylem fiber. No doubt about that. Then we'll move on to the phloem. Who is G? G is a companion cell. I'm sorry, F. F is like a bit of a, F can have a bit of a filtration, or a, it's more, let's say it is not exactly more open like a perforation plates. Things don't go very easily through the F. What do you mean by F? It's a sieve plate. No? F is a sieve plate. E, F, G. Then H. H is what? You can call it a sieve tube element. H is a sieve tube element. I, what are I? I are again pits. Okay. Then J is a fiber. Okay. Also, I volatility, if you have written if you have written plasmodesmata, I would give 1 mark, but not very correct. Okay. Anyone who has written I will to plasmodesmata here? Have you written? Yeah, I saw that. Yeah, you can take as plasmodesmata for I. It would have been more accurate. The plasmodesmata are actually located between the companion cell and the sieve tube element. So they are not usually located entire surrounding. But anyway, you can take marks if you have written those are as plasma day smart. No, no, you have to write vessel element, xylem vessel element, CO tube element. Those are the proper technical terms. Are they? You have to write those. Right. So that's it. Question number 3 done. Any questions? The labeling is clear, right? You can ask any questions that you have. Nobody is sleeping, right? Okay, shall we move on? Okay, let's move on. Any questions? Okay, yes. For parthenocarpy and parthenogenesis, how much? Okay, all right. Forgot to give you the marks. Okay. Make it for this labeling, there's 10 marks. Okay, for the 10 points. And then for xylem and phloem කියන එකට there's 2 marks. Okay, so yeah, මෙතෙන්ට 2යි, මෙතෙන්ට 10යි. Okay, and here for this whole list post fertilization changes, there's 5. Okay, and then for the all the parts, there's 9 marks. So, which one didn't I give? Ah, yeah. For parthenocarpy definition, 1 mark, and parthenogenesis 1 mark. Okay. Okay. Let's move on to fourth structured essay question. So I didn't plan to leave this much space for this question. If a person has given a name of a species, so in order to honor him, how do you include that in the scientific name? How do you include that in the scientific name in order to honor a person who has probably discovered that species, but you sometimes name species to honor some other person as well. Okay, so you have to say like this: it is indicated by a capital letter, okay, as abbreviation of a full word, of as an abbreviation of a full word at the end of the name. And it is not Latinized. Okay, so it is indicated by a capital letter. It's the abbreviation of a full word at the end of the name, and it is not Latinized for 3 marks. Okay, make a very event, right? It's just something. Okay, right. Name 2 classes of phylum Chordata that show. Who's drawing on my board? Okay, phylum Chordata. That which show of the each of the following features. Okay, endothermy, that means you are warm blooded. Endothermy means that you are warm blooded. Okay, who are the warm blooded people? You have birds, you have mammals. They are asking the classes. Okay, so you have to write the proper class name, right? Not mammals, not birds. You write aves and mammalia. I saw some papers where they have written mammals and birds. Okay, they are asking classes. Okay, keratinized structures are present in who? So they are asking only 2, ne? But if you take reptilia, you have keratinized scales. Then of course you have aves, you have keratinized feathers, and also mammals. You can have keratinized hairs, nails, and the skin epidermis is also keratinized. No, skin epidermis is also keratinized. Next question. External fertilization. Who shows external fertilization in Chordata? You have Osteichthyes. Bony fish. Bony fish usually have external fertilization. Have you watched Nemo? Nemo Balladino, you can remember there's a very old movie called Nemo. We're not very old, right? For you, bit old, I think, right? You might remember in the beginning of that movie, you have a lot of fish eggs. It's about story about some fish, no? So you can see a lot of red color fish eggs on the ocean floor before the movie starts, and all those eggs are destroyed. So the parents, the 2 parent fish, are discussing about their eggs, about the future of their eggs. So if they have fertilized eggs on the outside, that means what? They have done external fertilization. No, the female has produced the eggs, and then the male has deposited sperm on the eggs, and the eggs have gotten fertilized. Okay, so the fish in that movie are clownfish. Clownfish are orange and white stripes in a fish, right? So they are bony fish or stichies. So that's why they have external fertilization. And who else has external fertilization? Amphibia, right? Usually frogs also. The female frog lays down a sort of a layer like it's like a jelly of eggs. They are all the eggs are connected by a sort of a mucous membrane, and so they sort of floats. Either frog spawn, again, frog spawn. It's like you can see it on the sort of edges of these ponds. Ponds edges you can see frog eggs, and the male comes and releases the sperm on top of that. So amphibians and osteichthyes, they have external fertilization. Is it a good thing? Is it a good thing to do if you are trying to live on land? If it's is it a good terrestrial adaptation? What do you think? Is it a good terrestrial adaptation? No. Why? The eggs are outside. Outside means they can become damaged, susceptible to susceptible to desiccation. Okay, so it's not a good feature to have. Right. Okay. State three structures of a shark that can be seen externally, which are characteristic features of chondrichthyes. They're more and what are the things can you see? Mainly, we'll start with their tail fin. The mehemai. Have you seen a shark's tail? It's more pointed towards the top, and there's a small part at the bottom. If you take a normal sort of a api kana nikan sale kari wakari hama kana kelavalle kage moge kari you can see the tail fin is like that, no? So three structures, okay? So you can start with the caudal fin. Caudal means the tail fin is not symmetrical. So it's, you can see it is not symmetrical. Caudal fin is heterocircle. Then operculum is like a bony flap that is covering their gills. Okay, I'll show you. Okay, in the evolution tube, we had a osteichthyes and a chondrichthyes, right? Hmm. Maybe. Right. Can you see? Right. Okay. Medine. So this is the operculum. Operculum is actually, have you seen Amma Malu Suddhakandu Podagilla Balana? Right? When she's cleaning fish, right? You might have seen when she's removing the gills, she has to sort of pull and remove this sort of a bony part that is sort of hard. It's like hard like a shell. There is the operculum. It is the bone. It's a bony flap that is covering and protecting the gills. And also, I can show you the caudal fin in the osteichthyes, which is homocircle. Okay, homocircle or symmetrical. Then if you also you can see here, this is called the swim bladder, which is helping the osteichthyes to maintain their buoyancy. It helps them to float. Okay. And if you go to the shark, you can see the caudal fin is somewhat asymmetrical. So it's called heterocircle. And you don't have a swim bladder. You don't have an operculum. And the scales, of course, you know the differentiation of scales, no? Right? So you have placoid scales. You have placoid scales in your chondrichthyes and tenoid or cycloid scales in osteichthyes, right? Scales are different. So you can see these placoid scales are like teeth, no? The tenoid or cycloid scales are like, you know, the basic structure of bone, right? Right, the osteon. So it is kind of similar to the osteon structure, right? So these are the differences that you need to know. Okay. The no operculum absent, so you can see operculum absent. Body is covered in placoid scales. Like shark, dry fish, you can see. Dry fish, more shark, dry fish, you can see. You can take a piece of shark, dry fish, and check the scales. They are like pointy ends. You have seen normal fish scales, they are sort of like sort of spherical, sorry, round shape. No, these are like pointy, okay, placoid scales, and those are the common things. You can mention about their cloaca, but it is not so. Cloaca means it's a common chamber where the digestive system, excretory system, as well as the reproductive tract open. Cloaca. Okay, cloaca again you can talk, but it's not very visible to the outside. But I would have given marks. Okay, the reproductive tract, the digestive tract, and empty into the cloaca, a common chamber that has a single opening. So any three, I would give 3 marks and make it. Sorry. So for this previous one, so 2 marks each, 6 total marks. Next question. Name 3 phyla which include animals that do not possess respiratory structures other than body wall. Right? So, other than the body wall, they do not have anything. So, of course, uh, you or everyone, I mean, so even the simplest one have the body wall. So, they don't possess any specialized respiratory structures. Ne? So, your body wall is not a specialized respiratory structure. So, what they are asking is they do not have any specialized respiratory structure. So who doesn't have? Yeah, 3 phyla that do not possess respiratory structures. You can say Cnidarians, Platyhelminthes, nematodes, and annelids also. Ne? Wait. You can't have annelids. Ne? Hold on. Do annelids have a respiratory structure? Let me just double check. Do annelids have a respiratory structure? Yeah, you can't take. Yeah. Annelids can bear. Okay, annelids can bear. Yeah, these are the things. Cnidaria, Platyhelminthes, and nematodes. Okay, yeah, because in annelids you have parapodia. Parapodia, they behave like gills. Yeah, that's good. Okay, parapodia, they behave like gills in marine annelids. Okay, in marine annelids. That's good. Okay. So these are the 3 phyla: Cnidaria, Platyhelminthes, and nematodes. So 3 marks. 3 photosynthetic pigments that are present in both brown algae and golden brown algae. Yeah. What are the common ones? Of course, everyone has chlorophyll A, chlorophyll C, and I think xanthophylls. Those are the common photosynthetic pigments for brown algae and so rhodophytes and phaeophytes. Okay. So who are known as brown algae? So who are known as brown algae? Can anyone tell me who are known as brown algae? There is actually, what is the example that you have? Sargassum, Ned? Sargassum. Sargassum is the example for brown algae. Yes, very good. And who are known as golden brown algae? That's actually diatoms. Okay, diatoms. Because your gelidium is red algae. Yeah, very good. Your gelidium is a red algae. You can note down. Okay, gelidium is a red algae. So rhodophytes. Sargassum is your brown algae. We call them phaeophytes. Don't need that. Okay, and diatoms are your golden brown algae. So you have chlorophyll A, C, and xanthophyll tama. Those are the common things. Okay, good, good, good. Okay, 3 points. Right? How do the xylem vessels of spring wood structurally differ from the summer wood in plants? So the summer wood is designed to do what, and what is the sort of the what are they prepared to do? So summer wood is designed to just get through, just for survival. The summer wood is designed for just for survival, so that the plant can stay standing up. It has to just get through this. The spring wood is designed to facilitate the growth. Okay, so keep that in mind. Summer, you are just helping the trees stay up, support, and you are just going you are just getting through the difficult time. Spring wood, you are happy and the plant is happy, so you are facilitating the growth. So you can say, maybe there are several points you have to say. So when you are talking about the spring wood xylem consists of xylem vessels with large lumen and thin wall. Large lumen and thin wall. That means if you draw it, right, you can draw them. They have bigger. Their inner opening is larger. Okay, okay, and they have thin walls. If you go to the summer wood, you have thick walls and narrow lumen. Thick walls and narrow lumen. So why is that? Why are you having a thick lumen? Sorry, thick walls and narrow lumen. Mehemane. So that's why the summer wood appears darker. Summer wood appears darker and the spring wood appears lighter because of two things. Can you understand? Because the spring wood has thin walls and a bigger lumen, it's more sort of transparent again, because it's sort of the solid part is less. And if you go to the summer wood, it has thick walls and small lumen. Therefore, it appears dark. Okay, so right. Compound used to stain a cross section of secondary thickened dicot stem. They are again asking about this lignification. Okay, so I was planning to remove this question, but it's I somehow have forgotten it, right? Safranin, but you can take all marks for this. Okay, safranin is the chemical that you use to stain lignified plant parts. Okay, lignified parts. How does phloem sap mainly differ from xylem sap? How does phloem sap differ from xylem sap? So you have to basically talk about phloem sap. It contains sucrose 30% by weight. It may also contain amino acids, hormones, minerals lian amarui, but I wouldn't have given, I mean, I just common negative marks. Then, right? Minerals are found in both xylem and phloem. Okay. So amino acids and hormones are only found in phloem. So those are the only differences. Okay, understood? Okay, moving on. What name the element that is involved in signal transduction? That is actually common for our human body as well, because when we learn about the nervous system, you will understand calcium is important for our synapses, right? Synaptic transmission, where one nerve ends and the other nerve begins. There's a small gap, so that the signal transmission across that gap actually depends on this element as well. That is calcium. So in the plants also, signal transduction is by calcium. What is the structure produced from the ovary wall during fruit development in plants? Ovary wall looking at it, right? We talked about this earlier. We touched this. Yes, the wall of the ovary becomes the peel of the fruit, right? Yeah. What do you call it? Pericarp. Ah, yeah. So marks for this question. Phloem sap. Yeah, methanyl identity marks. No, right. Springwood and so there's 2 marks for the springwood. Large lumen, thin wall කියන එකට. Then summerwood, thick walls and narrow lumen, another 2 marks. So altogether 4 marks for that question. Okay. Then saffron වලට all ගන්න. Then this question 2 marks contains sucrose by 30% of weight, amino acids and hormones. Another mark. So altogether 2 marks. Calcium 1 mark. Pericarp 1 mark. How do plants detect gravity? How do plants detect gravity? By Ken. I would prefer if you have said by the settling of statoliths, not just statoliths. If you just have a statolith, what do you mean by a statolith? It's a dense group of starch granules, right? It's a dense accumulation of starch. But just because you have dense accumulation of starch, is it going to detect gravity? No. It has to move. It has to move downwards on the root, so that on the root cap, to be more accurate, right? So this is the root cap. So this is the root and this is the root cap, right? So in the root cap, your statoliths have to settle downwards, right? Your statoliths have to settle down at the bottom of the root cap in order to send a signal. In order to send a signal. So it is actually the settling of statolith. So therefore, I have given 1 mark and another mark. So altogether 2 marks. Okay. How do plants detect gravity? It is by the settling of statoliths. By the settling of statoliths. Right. Finally, final part of SEQs. Okay. How many ATP molecules are synthesized when one NADH molecule and one FADH2 molecule are oxidized in the electron transport chain? Very simple things. So from one NADH, you get 2.5 ATP. From one FADH2, you get 1.5. Three products of the link reaction of aerobic respiration of a glucose molecule. What are the products? Acetyl CoA, you get a carbon dioxide, okay, and then NADH, okay. But I would prefer if you have written the numbers 2 acetyl CoA, 2 CO2, and 2 NADH because they're asking a glucose molecule, but I didn't reduce marks, okay. But I would have preferred if you wrote the numbers as well. 3 marks for this, 2 marks for this. Based on fossil records, how many years after the origin of prokaryotic cells did eukaryotic cells originate? Yeah. How many billion years? Yeah. What is the timeline? So the protocells originated how long ago? Prokaryotic cells originated 3.5 billion years ago. Then eukaryotes evolved 1.8 billion years ago. So what is the difference? What is the difference? How many billion years gap? Yeah. What is the gap? Okay. How many billion years was there in middle from prokaryotes to become eukaryotes? Yeah. 1.7 billion. No? 1.7 billion years. So you have to remember both in order to be able to answer this. Name the group of organisms, animals, from which the earliest tetrapods, kapul hatarak tiyana, the organisms with four legs, four limbs, evolved from lobed fin fish. Right? You have might have seen memes about fish. Where have you been, Kilagwam? Another fish says, I have been to a jog. So I have been on a walk. The other fish ask, what is a walk? You would, I mean, you don't know because they haven't evolved yet. Okay. So the lobed fin fish, uh, it happened over many million of years, right? Lobe fin fish gradually sort of became more amphibious. They became more amphibious, so they have started to have a double life on land and on water. Then they gradually evolved into reptiles. Okay, so that's how the evolution happens. Appearance of food, first food chains. Make it I had to give all because it's not given in the resource exactly. Okay, first food chains take as all. I think all marks were given as all. And differentiation of large trees into roots, stems, and leaves, 380 million years ago. MYA. Okay. Right. Any questions? Did I go too fast? No, right? We started exactly around 5:15, right? So we were able to go through all four structured essay questions. Okay. So for the essays, okay, essays, I plan to give you the marking scheme, and also there has been request to discuss the last two essays, particularly the short note. Okay, particularly the short note. I will discuss that. Okay, so one of the short. What are the two short note? What are the short note questions? You have K plus influx, respiratory quotient, and soul stress. Okay, I think the secondary growth one you have written well. Sorry, hold on. So Calvin cycle one was written very well. Kingdom fungi was okay. The primary structure was very well written, everyone. And this one was not very well done. low temperature, cold stress. That wasn't very well done. So I will discuss that. Secondary growth was actually good. Most of you have written very nicely. Here, soil stress was a bit doubtful, but the other two structure essays were okay. Okay. So what I'm going to do is I will discuss the stressors. Okay. I will discuss the stress parts. Okay. But I will stop the discussion for now today because I'm fighting a bit of a fever. Okay, but we managed to discuss all the structured essay questions. So I will discuss the two stresses that is important. But you can inform me if you want any other personalized I mean, if you want myself to discuss, but I'm going to be sending the marking scheme for all essays, all four essays. So you can cross check with your marking schemes how you have written and how I expected you to write. Okay, so we'll stop the discussion for now. Any questions that you want to clarify? Anything? So keep joining with the paper class. The paper class is perhaps more important than the theory class. Okay? Perhaps more important than the theory class. Because theory you can self study, but papers, I know it's a joint discussion. We are going ahead with like a discussion based method. That is the most active. I mean, that is one of the most productive ways because there's an active recall. There's a question, we look for the answer, we think of the answer when you write the answer. So there's a lot of active things that you're doing that is going to help you to memorize things in the long, sort of keep them in your long term memory. That's what you want because no matter how much you think, you cannot memorize the entire bio syllabus within 1 week or the night before the exam. That is not going to happen. You might know this by now, right? Grade 12 third term test, did you have it? Not yet. Second term test you had. Third term test, did you have? Third term test? Anyone who had the third term test? Okay, but at this point you are entering into the fate sort of the stage where the syllabus is too hard to handle, right? So the only way you are going to be able to take all this information to the exam if you have them properly organized and also in your long term memory. Otherwise you can't take them with a just cramming. තඩබඩ ගාලා පාඩම් කරලා කලින් දවසේ night එකේ cramක් දාලා you can't go to the biology paper, right? That is not going to work. That is just going to stress you out. Okay, so the best way is through a repetition and also doing questions. You have to recall your knowledge again and again and again. Okay, right. If you have any questions you can ask, then otherwise we'll stop for the day. Okay, any doubts? So well done for the people who scored the highest, and of course we are going to have further more exams, and you can try to be on the top list. Okay, what matters at the end is the exam result. Okay, this is all preparation, but the preparation is the thing that is going to matter at the end. So keep working hard. Okay, then good night. Okay, welcome.